Okay, Jasmine, this is for you. In the center, we have a zwitterion. This is where we left last time, and it has a pH around 7, which is neutral. On the left-hand side, when I add acid, I'm adding hydrogen, so the hydrogen comes on and is back on the acid side, so it's a positive ion. If I do a base solution, which is about 14 on the pH scale, anywhere between actually above 7 to 14, you actually end up going back and forth between the NH 2 and I believe in the C. So I've taken the hydrogen off, the C and the COO minus with the R up here and the H down here. Okay? So they can go back and forth. Again, this scale goes all the way here. So depending on where on that scale you are, depends on what kind of ion that you have. Okay, so the isoelectric point is the pH at which the amino acid has no net charge. This is usually the zwitterion because it has a positive and a negative and it's canceled. Don't take that as a negative. Okay, so it has a positive side and it has a negative side to cancel out, and that's known as the isoelectric point. We can actually use what's called electrophoresis to separate these ions, to separate where they are. And depend on where they're migrating, we know what state they're in. Okay, I am just finishing then with 9.4, which finishes zwitterions. We'll talk about them more. But remember that really important is your table 9.1. You're going to be going back to that a lot. And that's on page 335 in your book. So this has all the amino acids, and they keep going back and talking about them. They also have the short names for them and also how to say their names, which is important. Okay, so on to 9.5. If you want to look at what electrophoresis is, they do have a picture, which is really nice on page 342. And you can sort of see how it separates what they are. Okay, cysteine is one of the amino acids that's unique because it's the only one that has an SH group. What was S? Sulfur. So it has an SH group, which is a sulfur. And it will form what's known as a dimer. Okay, and a dimer is just when the two sulfurs bind each other. So because of that, what we end up with is it actually turns because the sulfur is on the R strand. And so as you're looking at it, you got your C, your NH2, your COOH, and you've got a carbon attached to that, and then your sulfur. And that sulfur, and the hydrogen's on this side, that sulfur will then bind to another sulfur of another, my brain is just not working very fast today, cysteine. And so you get what's called a dimer, okay, which is a disulfide bond. Okay, so you get that disulfide bond. And that's unique to the cysteine. Only cysteine can do that because it's the only one with a sulfur bond. Okay, and depending on oxidation and reduction, depends on if we keep the disulfide bond or if we don't. So if the disulfide bond is reduced, we lose it, and it goes back to individuals. If we oxidize it, it goes to the disulfide bond. Okay, the next one, peptides. We have to worry about, they talk about different kinds of peptides, dipeptides, tripeptides, polypeptides. They use the prefixes that we have before. Di meaning two, tri meaning three, poly meaning many. Okay, and what that is when we have a bond that is an R, C, double bound O, N, and then an R. So this one would also have a hydrogen on it. So when we form that bond, that's a peptide bond. And that bond is formed by taking one of our amino acids. Remember, our amino acids have the R up here, and they have an acid side, and they have an amine side, and then they have the hydrogen here. Well, it comes up to pass by this side, meeting another one that is over here, NH2, CRH, COOH. And what happens is one of those hydrogens coming off here and mixing with the OH on here and forming water. So that's how we get this bond. And one of those bonds is known as a peptide bond. Okay? Everybody good with that? So if we have a dipeptide bond, what does that mean? How many amino acids are in a dipeptide bond? 3. Okay, if it's a polypeptide, we have many. If it's a tripeptide, how many amino acids? 4. Okay, so you can see how that works. So in order to have one bond, we need 2 things. Okay, so let's see, see the top of page 345. And that's basically what we've done. You need to be able to recognize, looking at a long string of peptides, bonds, what those amino acids are. So you have to go back and say, oh, okay, this would have been split here. So what was this amino acid? What was this amino acid? Okay, does that make sense? Okay. All right. That peptide bond, as you go along, that's known as the backbone. And the things coming off are the side chains. Okay? All right. I'm not going to have you worry about peptide nomenclature. They start going into peptide nomenclature about page 346 and the rules for it. I'm not worried about the YL, the RL. I am worried about you being able to tell me in order what peptide bonds we've got. So in other words, if you're looking at page 347 at the top, in the practice exercises it says GL-ALA-LEU. I don't want you to be able to name it, I want you to be able to draw it for me. Okay, so taking them just like we did up here on that bottom and putting them together and showing me what it looked like all together. Okay, so let's see if we can do that. Let's just take A, which is ALA dash, whoops, that was the second part of it. Okay, try again. Oh, itty bitty eraser, I forgot I put an itty bitty eraser. So GLY dash ALA dash LEU. This is where I said that going back to table 1.1 is going to be important. First of all, let's write what those are underneath here. And we'll always put the acid side on the right just to help us out here. So GLY. Where is it? Have we found it? Oh, it's right up the top. Okay, so we got NH2 dash CH HCOOH. Okay, what's ALA? Right next to it, right? So we got I still want to do N backwards. NH2 CH, whoops, CH3 HCOOH. And then we've got LEU, which is H2NC CH2 CH CH2 CH2. Or is that 3? I can't read it. It's 3. It should be 3, right? And COOH and H. Okay, so all I've done is take them right from the table. So now what we got to do is we got to take the water out to make a peptide bond. So one hydrogen in the OH, one hydrogen in the OH. So as I do that, I'm going to rewrite it. H2N C H H C double bound O N H C CH3, H, C, double bond O, N, H, C, CH2, CH, CH3, CH3, H, COOH. Okay? Do you see how I did it though? Okay, just take that water out. Take one hydrogen from here and one hydrogen from here, and then I just smashed it together. I took the water out and dehydrated it to make the peptide bond. I will probably copy that chart for you for the test. Okay? All right, so you need to be able to just transfer this down here. Take this part down here that doesn't want to right now. This part right here, okay, it's not going to write for me. The bottom part in black to the A, now it's writing for me. To this part right here, you need to be able to go back and forth between this and that. Okay? And you need to be able to go back and forth between the zwitterion and what it is an acid and what it is in base. Are we okay? That's all I'm going to teach you new today. Okay, your assignment's on the board. I'll write it up here too. Do you need me to go back? Sorry.
Yeah.
Okay. Whoa, hello. It really wants to not go to that page anymore. There we go. So it's on the blog, but it's page 373, number 28, 30, 36, 41, 42, 44, 46, 50, 52, 62, 64, and 66. So you can get that assignment from the blog, Jasmine.